So with that, I just want to thank the people involved. I highlighted Stefano and Lara. I also want to thank Wanda and Patrick. We did a nice selfie here in front of the electron microscope. I also want to thank the rest of my collaborators and my funding, and thank you.
The second lecture in the Neuroscience Bridge session is delivered this year by Dr. Don Cleveland and is titled Development of Designer DNA Drug Therapy for Neurodegenerative Disease. Dr. Cleveland is currently chair of the Department of Cellular and Molecular Medicine and the Distinguished Professor of Medicine, Cellular and Molecular Medicine and Neurosciences at the University of California at San Diego, where he is also the head of the Laboratory for Cell Biology in the Ludwig Institute for Cancer Research. Dr. Cleveland graduated in physics from the New Mexico State University, and he received his PhD in biochemistry from Princeton University. He did then postdoctoral work at the University of California, San Francisco, and he became a professor of biological chemistry at Johns Hopkins University School of Medicine in 1981, moving to the University of California, San Diego in 1995. Dr. Cleveland has made multiple pioneering discoveries in biology, neuroscience, and in cancer biology. Among these, I'd like to mention, first of all, his discovery of and characterization of tau, the protein that accumulates abnormally in the neurofibrillary tangles in the patient with Alzheimer's disease and in many other forms of tauopathies, as we now call those diseases. He has also made important contributions in the understanding of the mechanisms of neurodegenerative diseases, particularly amyotrophic lateral sclerosis, as well as in the regulation of the movement of the chromosomes and the mechanisms of mitosis, cell division in normal cells and in cancer cells. Furthermore, Dr. Cleveland has pioneered the use of DNA antisense oligonucleotides, which he termed designer DNA drugs, for silencing of target genes in the nervous system, and for their use as a therapeutic approach in several diseases of the nervous system. The antisense oligonucleotides approach has undergone an exponential development, leading to several ongoing human clinical trials. Dr. Cleveland has been publishing extensively in top scientific journals, and he has received many awards, including the election to member of the US National Academy of Science in 2006 and the National Academy of Medicine in 2012. For his development of the designer DNA drugs approach in 2018, he received the Breakthrough Prize in Life Sciences. It is therefore a great pleasure to give the word to Don Cleveland for his lecture on development of designer DNA drug therapy for neurodegenerative disease.
Thank you very much for that very kind introduction and for the opportunity to present at the Neuroscience Bridges Lecture at the International Parkinson and Movement Disorder Congress. What I'd like to describe today is our development of a designer DNA drug therapy for neurodegenerative disease. The story began from the realization that the genes whose mutation or altered expression is causative or contributory to the major diseases of the aging nervous system, Alzheimer's, Parkinson's, ALS, Huntington's. All of those genes are widely, some ubiquitously expressed, not uniquely expressed in individual classes of neurons whose dysfunction gives rise to the disease phenotype. Moreover, it was the discovery of Andy Singleton and his colleagues in 2003 that inheritance of an extra gene copy, normal gene copy of the gene encoding alpha-synuclein is causative of early onset Parkinson's disease. And that was followed by a similar discovery that triplication of the gene encoding amyloid precursor protein with no mutations, just the normal gene, but 3 copies of it, is causative of early onset Alzheimer's disease. Recognizing that, an on-disease mechanism therapy would be to lower the synthesis of those genes, either of those genes, in at least some of the cells that express the gene in ways that drives disease progression. So we set out to do that. And for the young folks in the audience, I know all of you would say, well, let's use an SI or an shRNA. But we chose an even older approach. using a single-stranded modified DNA, which if you can get that DNA, which I call a designer DNA because we design it for a single gene target. If you can get it inside the target cell so that it can diffuse in the target cell nucleus, it can pair with the pre-mRNA target, making an RNA-DNA duplex, the RNA of which is a substrate for an endogenous, nearly ubiquitously expressed intranuclear enzyme RNase H that cleaves the RNA, but not the DNA, which is then freed to go catalyze degradation of another RNA target. And over the years, the medicinal chemists have modified the phosphate backbones and the 2' and 5' positions of the ribose sugars to produce antisense oligonucleotides, or designer DNA drugs, which have remarkable properties. You know, when you take an aspirin, you typically get 3 to 4 hours worth of relief. But with these modified DNA oligonucleotides, we don't get 3 or 4 hours. We don't get 3 or 4 days. We don't get 3 or 4 weeks. We get 3 to 4 months worth of efficacy from single doses. Oh, but one other feature of this class of drug, when administered in the periphery, either orally or by injection, none of it gets into the nervous system. So in the examples I'm going to describe today, we're going to directly inject it into the fluid that bathes the brain and the spinal cord, the cerebral spinal fluid. And it's from that kind of introduction that we get efficacy, as you'll see, broadly throughout the nervous system. The disease we started with is amyotrophic lateral sclerosis, or ALS, the salient feature of which is the selective degeneration and then death of the upper and lower motor neurons. The paths of these neurons were noted more than 100 years ago by the great neuroscientist Ramon y Cajal, and here's his drawing as the upper motor neurons send their axons down the spinal cord and synapse on the lower motor neurons, well, directly, as he's drawn it here, or more frequently, indirectly onto the lower motor neurons, whose job it is to innervate the muscles and trigger their contraction. In ALS, it's a late onset disease, almost invariably fatal over a time course of a handful of years. And your lifetime risk of dying from this disease is between 1 in 1,000 and 1 in 2,000. And a simple calculation therefore is with the current world population, there are something on the order of 5 million people alive today who will die from this disease. About 10% of disease is dominantly inherited. And indeed, this is the founding paper for the first mutation that is causative of inherited ALS, led by Bob Brown and his colleagues worldwide, who identified that mutation in the gene superoxide dismutase, a ubiquitously expressed gene, is causative of a proportion of inherited ALS, and it accounts actually for about 2% of overall ALS. Over the years, a consensus has emerged that these gene mutations cause disease through acquisition of a toxicity from the encoded gene product, not through loss of function of the normal gene. And although the mutations were identified in 1993, it's a little sobering that in 2021, we as a community still don't agree on what the toxicity is. But we could be directly on disease mechanism if we could just turn off the disease-causing gene. And here's the founding example of doing that, of a therapy attempt for a neurodegenerative disease by using a designer DNA drug in the mammalian nervous system. And here we're targeting the causative SOD1 mutant RNA level with a designer DNA drug, an antisense oligonucleotide. And you can see here, near the site of injection of the drug, we can lower the level of the RNA, the mutant RNA. Actually, we can do so, lower it almost to 0 in a dose-dependent manner. Here, we've lowered it to about 40% of the original level. Oh, actually, and somewhat surprisingly, farther away from the site of injection into the cerebral spinal fluid, we lowered it. Actually, we lowered it throughout the entirety of the rodent nervous system. I should have mentioned, this is a transgenic rat that expresses an ALS-causing SOD1 mutation. And when we dose those animals at disease onset with this SOD1 targeting antisense oligonucleotide, well, this is what we saw. If we mock dose them or dose them just with saline, the animals, it's a hyper-aggressive model of disease, and the animals live only about 25 days after initiation of symptoms. But when we dose them at onset with an SOD1 targeting oligonucleotide, we could double survival after onset. So we argued from that this antisense oligonucleotide therapy for neurodegenerative disease was a plausible strategy for not just for this example of inherited ALS, but for many examples after we demonstrated that indeed, both in rodents and in non-human primates, that we could deliver these drugs almost universally throughout the nervous system from focal injection. Okay, so, and indeed, just last summer, here's the outcome of a phase 1, phase 2 trial targeting this mutated SOD1 in inherited ALS, where the team showed that they could do so safely, but even more so, if you look over here, this is a clinical efficacy on the vertical axis. And if we compare patients that had disease, very rapidly progressive disease from the most aggressive SOD1 mutations, those are in the blue dashed curve, the placebo-treated patients continued to decline over an 85-day course, but the treated patients, the disease nearly stopped. And that was also true of the overall cohort of patients with mutations that gave to slower progressive disease. So from that led to a phase 3 trial with a much larger cohort of patients, and we're eagerly awaiting the outcome of that phase 3 trial, probably at the end of this year. A second example was in the childhood disease, spinal muscular atrophy. It's one of the leading genetic causes of mortality, inherited causes of mortality in children. The feature of it is that in patients are homozygously inactivated for one gene, the SMN1 gene, a protein that's involved in pre-RNA splicing. And in its severe form, it's fatal in the first year or two of life from paralysis, from never having generated motor neurons that could successfully innervate muscle. And here's the genetics of the disease. The patients have homozygous inactivation of the SMN1 gene, but we, almost alone among the primates, have a second copy of that gene, SMN2 gene, but unhappily, there's a single base change in that gene so that it is misspliced to miss out exon 7 and make a non-functional protein. So the strategy was to use an antisense oligonucleotide